All right, so thanks for having me here today to speak. I'm a postdoc of Pepe's, and unfortunately she couldn't be here today, but she says hi. And the title of my talk is Regulatory Principles of Cardiac Gene Expression on a Global Scale. So Pepe's lab is actually more of a proteomics lab, and originally the talk was going to be on deep learning of like post-translational modifications in mass spectrometry. But unfortunately, since she couldn't be here, you're going to have to listen to me talk about the double injury model for about 15 minutes. All right. So just a brief overview of the talk I'm going to give today. First, I'll be going over the double injury model and the UMAPs that we've been annotating, as well as the multi-omics pipeline that we set up for downstream drug screening. So how does this model work? So first, there's the first injury of apex resection at postnatal day 1. This is followed by a 4-week recovery period, after which there's a second injury in the form of an LAD ligation at P28. And then we explant the tissue latest at postnatal day 56, so this is 4 weeks after the second injury. And then we've also collected samples at P30, P35, P42. And samples were collected from the apex zone, the border zone, remote zone, and cutting zone. Okay, so just to give some background on the project, so during my PhD at UAB, we were working on this large animal model system, and basically what we found was that there was this conservation of the regenerative window shortly after birth in large mammals, similarly in rodents, but that this basically that this window is conserved. So then this led to the hypothesis that I originally thought of during the PhD, and which UAB gave me a lot of flack for because conflict of interest. My dad was a PI, so. But anyways, the hypothesis was that the injury-responsive neonatal cardiomyocytes are phenotypically immature relative to the developmental control cardiomyocytes, and as such it can be seen to represent a spectrum of cellular states with distinct gene expression signatures that differ from non-responsive, non-proliferating cardiomyocytes. So if this regenerative window is molecularly regulated, then the idea was that the second injury would be able to induce further proliferation, thus giving a larger signal that we can detect through single-cell sequencing. And so this next slide, I'm just showing how this, how the experimental groups that we have. So in the developmental controls, we have the control P1 cardiomyocytes. 4 weeks later, that control P28, and then finally, 4 weeks later, that control P56. So these are our 3 developmental control groups. We have the single injury in the form of apex resection at postnatal day 1. We age them 4 weeks, and then we collect the samples, and that's our single injury group. It's in the form of apex resection at postnatal day 1. And as well, we have the same sort of experimental design collected 4 weeks later at P56. There's a double injury, which is the apex resection at postnatal day 1, followed by a second injury in the form of LAD ligation at P28, and then we collect it, as I said before, at around P56. Basically, what I'm showing here is that what I'm hoping that you'll see is that there's these cellular states are each injury kind of causes like a sort of like matrix of cells with different gene expressions and different regenerative potentials. So if you look at it that way, it's a pretty good platform by which you can sort of model maybe cardiomyocytes in different cardiovascular diseases on the single cell level. And I'll tell you a little bit about how we set up this platform, and we're still in the process of making it. But finally, there's a double injury control, and what this group consists of is the P28 or an LAD ligation at a time beyond this proliferative window. So basically, we do an LAD ligation in the P28 animals, and then we collect it 4 weeks later. So those are the groups that we're working with. So then after we sequenced them, we had to cluster them. This is just showing by experimental group of what I've showed earlier. So what you see here is that this fetal sample in pink is pretty distinct. And I'll show you in the next slide that this little branch here is proliferating. And that's the fetal proliferation. You got the postnatal proliferation made up of all the different time points. And then the developmental controls are pretty distinctly mapped within this heterogeneous cluster consisting of various injury responsive cardiomyocytes. And then you got the control P56 here. After seeing the nice talks yesterday on the epicardial activation, I actually looked at the gene markers and I thought they were all immune cells, but actually it's like immune and epicardial. And then we have the skeletal muscle cluster popping up about a week after the second injury. And it might be like an experimental error, but it seems like there is some shared gene expression. And you can see it from here. This is one of the postnatal proliferating clusters, or postmitotic actually. So I'm thinking this might actually be some sort of signal, but we'll see. Okay, let me try this. Nice. So using a resolution of about 2.5, we have this nice UMAP annotated here with the different clusters, just showing that, yeah, these are distinct clusters with defined by distinct gene signatures. Okay, so when I said there's proliferating cardiomyocytes, it's actually 3 distinct clusters. So there's a prenatal, postnatal, and then I think there's a postmitotic right here showing up. And just to show you some characteristic genes that are enriched in these clusters, you get a lot of Ki67, kinesins, cell cycle regulators that are like transcription factors, topoisomerases, et cetera, et cetera. So just mapping it over time, these clusters, you see that it's pretty distinct for this fetal cluster 32. It's only showing up mostly in the fetal samples. Cluster 35, which is the ones that I think are actively proliferating, it peaks around 14 days after the second injury. And this is similar to what we've found in the PLOS1 paper that I talked about earlier. Basically, we're using bulk RNA-seq data and looking at like the gene changes over time post-injury. And we estimate it to be about 1 to 2 weeks, and we see that here. This 37 cluster is what I think to be dedifferentiated postmitotic cells, and they also express some satellite muscle markers, so it's pretty interesting. Okay, so I talked about basically just analyzing this UMAP, analyzing this UMAP and the single cell sequencing data, and for my PhD, and about maybe 6 months into it, Pepe is like, okay, Eric, you got to get off your dad's payroll and actually do something with like that I want to do. And so we pushed the project into this multi-omics direction. And the rationale behind integrating other omics data is, well, because you get re-expression of fetal cardiac genes in heart failure patients. So then if you view it this way, then each sort of cardiomyocyte within a heart at various stages can be viewed as individual events or cellular states. And by integrating this single-cell sequencing data with genome-wide association studies, as well as later we'll do proteomic and metabolomic studies, we can interrogate the underlying disease pathogenesis on the single cardiomyocyte level, with the big aim downstream to identify druggable loci or single cell expression quantitative trait loci to induce the phenotype that we want. And then once we set up this platform with, which right now consists of single cell data integrated with genome-wide association studies, the idea is that we can do deep learning cell lineage analysis on these sort of modules. So I had a couple of questions about, like, initially of whether there was an existing sort of analysis platform that we could use, and looking into the literature, I stumbled upon Diego's paper that he published while he's a postdoc at Stanford. Right now he's in Cole Trapnell's lab. But he published this paper, this algorithm called roly-poly, which does basically the exact thing that we were looking for. And he basically said that, yeah, this is kind of what you're looking for, but there's been a lot of work done since. And he directed me to this paper that's in Press in Nature Genetics right now, published by Martin Zhang and Kang-Cheng Ho. So Martin's at Harvard and Kang-Cheng's at UCLA. But basically what they do is they have a sort of algorithm that's able to take genome-wide association study or population studies, take the summary statistics from that, and from that data assign a disease risk score for that single cell data. So setting up the pipeline, what we have here is that we have the, in the single cell sequencing side, we have cluster phenotypes that were annotated by students at UCLA, the differentially expressed genes as well as the transcript count values. So you get like a bunch of information there. On the GWAS side, you have the patient phenotypes, their variants, and the location of the genes. And what you do is, first we use Magma to process summary statistics, extract the relevant SNPs, locate the bordering genes, rank them. And then SCDRS and Magma basically create this ranked gene list. And what SCDRS does is it integrates this into the single cell data, and then basically what we're aiming here is for a single cell expression quantitative trait loci, which right now it's open to definition, but I think we're slowly getting there. So in order to set up this pipeline, we had to use a different workflow. So we switched from using R to Python, I guess, and then we did some Leiden clustering versus Seurat clustering, and we see that this is what we get, and I haven't looked deeply into the annotations yet, but the patterns that I've shown earlier of the proliferating clusters remains the same, namely that there's one branch that is mostly prenatal, one's postnatal, but there'll be more work done on that. So this is the first single cell disease risk score that we got from the Hermes trial, or the Hermes consortium on heart failure. And you can see that the higher disease risk scores are more in the center of the clusters. I don't really know the significance of that, but what you do see is that these proliferating clusters over here, marked by these genes, and these are just a couple of them, have really low scores, indicating that it seems like these lower scores, not surprisingly, are associated with cell cycle progression and proliferation. Whereas on the other hand, the higher risk scores seem to be associated with early response stress genes, and then downstream of that you get higher expression. So I think there's like some sort of cell signaling pathway that's underlying these events. But that's going to be what we're going to look into next. So I'd like to thank Peipei Ping, my mentor. She's a great PI for me, and Ding Wang for helping me with these slides because, not my strength. As well as everyone in the lab, my undergrad students, who really, I enjoy working with them a lot. The Zhang lab, Yuji, Tan, Greg Walcott, who did all the pig surgeries, Zhang, and Dan Gary's lab, who helped us do some of the bioinformatics analysis on the circulation paper that just came out, as well as our collaborators, Diego, Martin, Kang Cheng, and Jake Chen at UAB. Thank you. I'll take questions.